Hi, in this lecture, we're going to learn more about the B cells and immunity. Now remember that the B cells are involved with antibody-mediated, or we also call this humoral immunity, because remember, antibodies are in your plasma, so they're in fluids. And so these antibodies are, again, specific or adaptive immunity. They attack specific antigens with these antibodies, and they're very, very specific. Antibodies only attack one kind of antigen. And we have millions of populations of B cells, and they're capable of making millions of different antibodies. So as we said earlier, you're going to definitely be able to recognize and produce antibodies against any pathogen. Now, the B cell's a little bit different. The B cell can directly bind to an antigen. It doesn't need the little cup holder, the little docking station that the T cell needed. So the B lymphocyte can actually bind directly to an antigen and start to recognize it. We say it's become sensitized once it binds to it. However, next it needs that helper T cell. So we talked about the helper T cell and the importance of that. Remember, the helper T cell has to become activated, as we did in a previous lecture. But once that helper T cell is activated, it then comes up to the B cell and it continues or finishes the sensitization. I can't even talk today. It activates it. We'll go with that. So once the B cell is activated, it's going to continue on its way and basically make clones. Now, one group of activated B cells become plasma cells, and the other group are going to become the inactive or memory B cells. So the B cell directly recognizes the antigen. A helper T cell releases cytokines that finish the process. Now it's sensitized or activated. It makes clones. One group of clones becomes memory B cells that hang out, and if you're ever re-exposed to that exact same antigen, those memory cells immediately become plasma cells. The plasma cells are going to make the antibodies, and the antibodies are going to seek out that antigen because they're going to be specific to just the antigen. Whereas, remember, the memory cells are going to stick around, and if you are reinfected, they will immediately become plasma cells and make the antibodies. Now, I mentioned that the antibodies are in the fluids, primarily in the plasma. They could be in the lymph, and you can measure your antibodies against specific antigens by doing a test, blood work, called an antibody titer. Now, I'm not going into a lot of detail on antibodies. I've mentioned before, they kind of look like the letter Y, and what they have are a couple different chains of peptides, of proteins. So we have a heavy and a light chain, and we have a constant and a variable region. What I want you to know is the constant region is kind of the stem of it, and then you have the variable regions, which can change, so they can change their shape to bind to different antigens. So the constant segment is how they're categorized, but that variable is very important because it can shuffle and change its shape so it can bind to the antigen. Now remember, when an antibody binds to an antigen, it forms an antigen-antibody complex. If we look at the classes of antibodies, we'll see that there's 5 classes. Now, we don't really do much on IgD. I just want you to know it exists, not really do much about it. We're going to focus on the other 4. So IgG, notice this is the most numerous. It's the majority, most numerous antibody that's in our bodies. It's in our different fluids in our body. They're listed in your notes. This is the antibody that can cross the placenta. So when we talked about issues with mother and fetus, it's IgG. And IgG is associated with what's called the secondary response. So this is the one that goes up when you get reinfected. So if I want to know if I have a COVID infection, if it's a recent infection, or if I'm having antibodies from like months ago. So you heard about people going to do antibody testing for COVID. If I go in and I have a lot of IgG antibodies to COVID, then I know that it's not recent. I know that if I have them, I was exposed. So if I have a positive test and I have IgG antibodies, this isn't my first time I got it. Now, I'm going to jump to IgM. IgM is the largest antibody, but this is the one that's released during a primary response. So I go in, I have COVID, I test my antibodies, and I don't have any IgG, I only have IgM, well, that's my first exposure. So IgM goes up for your first exposure, and then any subsequent exposures, it's going to be IgG. That's what's called the secondary response. So when we measure a titer, we're really measuring IgG antibodies. Now, IgE comes from, it really helps activate those mast cells. Let me cover that up a little bit. The basophils and the mast cells. So when IgE binds to a mast cell or basophil, that's what triggers the release of histamine and heparin, and it really causes a lot of inflammation to occur. IgA is found in a lot of secretions. It's found in saliva, it's found in your GI secretions, it's found in tears. And so this one is another one associated with basically a lot of different secretions in the body. Sometimes you'll hear about your gut is part of your immune system, and part of that is because of the amount of IgA antibodies that's released from there. So we're talking about these antibodies, but what's the big deal? What do they do? Well, first of all, I want you to understand that they do not directly attack and kill a pathogen. That's what a killer T cell does, right? And even NK cells and macrophages, they don't directly do it, but they slow everything down. They stimulate the immune system so that you can get over the illness. One thing they do is they agglutinate and precipitate. So what antibodies can do is kind of get together and kind of clump up a pathogen. And so they agglutinate, get in a clump, precipitate, crystallize. And so what they do is they can clump it together so that it can't infect your cells. We talked about the opsonization. Remember, that's like putting the grippies on the bacteria. So the antibodies actually attach to the pathogen so that it's easier for a macrophage to grab it. They neutralize things. We've looked at this a little bit with some other cells. So when they neutralize, they actually can bind to the virus, for example. So the virus attaches to the antibody, and then it does not actually infect your own body cells. So they work kind of like a decoy. They activate complement. Remember, complement was a protein that formed that MAC complex. So it's really important, and this is one of the things that starts that whole domino effect of complement. We also know they cause inflammation. Inflammation doesn't feel very good, but it's very important because it slows the infection down and prevents it from spreading. So it helps with the mast cells. And then it even activates your NK cells. So phagocytes and those natural killer cells are going to go to the area. So it basically slows down the spread of the pathogen and activates your defenses to fight the pathogen. So when we're looking at this response and memory, we talked about immunity at the beginning and competence, right? The ability to have an immune response. We have our primary and our secondary. So if you look at the graph here, notice the IgM levels go up in your first exposure. So it is produced a lot faster. But if you get exposed again, your IgG levels go up really high. In fact, a lot of times you form the antibodies so quickly and you have some of these antibodies so you don't even feel sick. Notice also the time it takes to get a good antibody response. A lot of times it can take a couple weeks until you get one. So when we look at an antibody titer, they're usually done to see your immunity to different diseases. They can be done to see, do you have immunity to COVID? Do you have a good IgG anti-COVID test? Sometimes the antibodies are used for diagnostic testing as well. We've talked about that, where they could bind to an antigen to show if the antigen is present or not. So we're going to go into types of immunity. So how do we have this ability to fight disease? Well, we can have two forms of it. So first of all, you're going to see something called innate immunity and acquired immunity. Now, innate immunity is just, you're born with it. It's just different diseases affect different species. So humans don't get distemper, dogs do. So we're not going to focus too much on that. We're going to focus on the forms of acquired. So it can be acquired through active or passive means. And what we're looking at with immunity is we're looking at, do you have antibodies? Because the antibodies are going to protect you. So active processes means that you have to make your own antibodies. So any example of active immunity is the individual makes their own antibodies. In passive, it's transferred. You don't have to make them. So, passive immunity, when are you given antibodies? Well, the best example of naturally acquired is breastfeeding. When a mother breastfeeds her baby, antibodies are in the breast milk, and that's how they are spread to the baby. We know some are transferred across the placenta, though a lot of them are going to happen in breast milk because babies don't make antibodies very much at all in the first year of life. Now, artificially acquired, or we would say induced, this is when you're given antibodies from someone or something else. So, for example, when COVID first hit and it was, we had high death rates and people who survived it had antibodies against it. And so you could take the plasma and spin it down and get those COVID antibodies and give them to a patient. And in a lot of cases, it saved people's lives. If you have rabies, a lot of times with rabies, they use horse antibodies. So they can get the antibodies against rabies from a horse and inject it into you to help slow it down. So again, in passive, you're not making the antibodies, they're given to you. In active, you gotta make them. So active, naturally acquired, how does that happen? You get sick. You get exposed to it. So you catch COVID, you develop antibodies. You get the cold, right? A flu. So you get exposed to the pathogen and your immune system does what it's supposed to do and it makes the antibodies. Okay, well, how do you get the artificially acquired or induced? Well, that's when you get a vaccine. So the only difference between actually getting COVID and getting a COVID vaccine is just, your exposure to it. If you get exposed to the vaccine is going to target and basically not hit you with a high dose of the pathogen. The idea with a vaccine is that it's giving you a small dose of the pathogen so that you can go through that whole process. Both of these, you got to go through the B cell. The B cell has to recognize the pathogen, right? It has to recognize it, whether it's the pathogen in your body or from the vaccine. And then you have to make those plasma cells and you got to make the antibodies. So once you get those, though, remember, now you have antibodies, now you have immunity. So I'm not going to go into a lot of vaccine stuff, but I do want you to understand kind of how they work. So some of the earlier vaccines, they just gave you the pathogen. They tried to attenuate it. So attenuated means they weakened it. So maybe they took out what would make it pathogenic. And so you got exposed to it, but it was weak and it should not have made you very sick. They also can inactivate it. Sometimes they kill it. So they might kill it in formaldehyde and they might kill it in something and give you the killed version. Again, the idea is it can't replicate. When you see this thing called adjuvants, this is one of the things that people who talk about vaccines say that this might be why they're worried. I mean, vaccines just mimic your own body. But an adjuvant is something added to kind of make sure your immune system recognizes it. And so one of the things they've gotten flack for is some had mercury in them, and there's different things added. So that's the piece that I think a lot of people might question a little bit as far as what's actually in the vaccine. And you have a right to read the vaccine information and see what exactly is in it if it has an adjuvant in it. Sometimes they just give you a subunit. Sometimes they might take just a piece of it, like the spike protein from the coronavirus, right? If they could just expose you to the spike protein, you shouldn't get sick because you're not getting all that viral genetic material inside your body. So sometimes they'll do a subunit vaccine because it's safer. And now we have the DNA and the RNA vaccines where now they just take the actual DNA, inject it in through the vaccine, and then your body cells pick it up and they are going to make that particular protein that way. So there was a lot of questions about the bioengineered, the DNA, the RNA. So I'm just going to show you the mRNA. This was the more popular, right, vaccines that we have out. And so the cool thing about the mRNA is that you're not getting the virus. You're not getting a piece of the virus. You're not getting the viral DNA. What you're getting is messenger RNA. So you're getting the messenger RNA to how to build the spike protein. And messenger RNA breaks down fairly quickly. So once it's injected, that was an issue with the vaccines, how to store them. It goes in your cell, messenger RNA. The messenger RNA enters a cell, and then the ribosomes use the messenger RNA to make the spike protein. It doesn't go in your nucleus, doesn't need to, and it gets broken down. So the messenger RNA tells your body cells, your ribosomes, how to make the spike protein. There's your antigen. Now you're going to get antibodies and get an immune response. So that's why they are used. They seem to be a lot safer, and they can make them quicker because it's just messenger RNA that's being used. So we're looking at an overview really quick. I only like this picture because it kind of shows you the sequence. I have to break everything down in pieces, but you get sick, right? You get exposed to something or you get a vaccine. So the very first thing, remember, is the neutrophils, the NK cells. Notice that our non-specific, our innate, is the first to respond. And remember, by the time you feel sick, these guys have been pumping along working. You usually don't get symptoms till about 24 hours after you're exposed to something, 24 to 48 hours. So, sad to say, you've probably already been spreading it to everyone the day before you knew you had anything. But then we have our adaptive, right? We're going to get our cytotoxic T cells. They're going to take a little longer, our plasma cells. So when we're looking at our specific immunity, our adaptive, when we get T and B cell, that we're looking at probably 4 days to 2 weeks before we get a really good response. So sometimes you'll go in and get checked out and they'll say, well, just give it 2 weeks and see if you feel any better, because it might take 2 weeks for you to get a really good immune response to it. And I'm going to wrap up this one. I know this is a busy picture, but I really like how it puts everything together. So we get exposed to some sort of antigen, right? We trigger our defenses. So the first thing that happens are the innate defenses. It's broken through our skin. So we have our protein and we have our cell. So those happen first. Then we go into our adaptive, and this is a nice summary showing you your MHC class 1 and class 2. So the little class 1 holds the antigen out to the CD8, the killer T cell. When the killer T cell is going to be activated, the T cell receptor and the little CD8 co-stimulator, right? We're going to have our activated killer T cells. They're going to go kill. We're going to have memory T cells that hang out and wait. If they're ever re-exposed, they immediately become the killer Ts, not going through the suppressors. Then when we have our helper T cells, they use the CD4 co-stimulator. So sometimes we call them the CD4 cells. They're activated by the MHC class 2. When they become activated, we have active helper Ts. They release lots of cytokines and cause lots of very important immune changes, antibodies, activating macrophages, activating killer T cells. And then we always have some memory ones that hang out. Once the B cell gets activated, it makes the antibodies. And then again, we still have that memory. We have the memory B cell. So if you're ever re-exposed, they immediately become plasma cells and they make those antibodies. So those antibodies are directly made from a plasma cell. A plasma cell is a cloned activated B cell. So hopefully trying to put it all together this way is helpful to you, and that finishes our lecture on the B cells.